Hello students, good morning to all. In this session, we are going to take up a new concept from the chapter Molecular Basis of Inheritance, that is Human Genome Project. But before going to Human Genome Project, let us try to understand the term genome. What is genome? Genome is defined as the total number of genes located in haploid set of chromosome. That is, in human beings, there are two sets of chromosome and they are called diploid. 2n is equal to 46 in human beings. So now the genes present in n number of chromosome, that is 23 chromosomes, or we can tell the total number of genes present in a gamete, because gametes are haploid. So therefore genome is nothing but it is the number of genes or the total number of genes present in haploid set of chromosomes. Now let us try to understand the term genome was when it was coined. That is, the term genome was coined by H. Winkler in 1920. Now let us go for the definition of Human Genome Project, HGP. Very important abbreviation. So let us define what is HGP. It is an ambitious international project designed to understand the genome of a human, that is, to identify and locate 35,000 to 40,000 genes, or you can tell 3 into 10 to the power 9 base pairs present on 23 pairs of chromosome. That is, even though we say genome is genes present in 23 chromosomes, but when we consider human being, when we know 23, the next 23 chromosomes will also be known. So therefore, we say it is 23 pairs of chromosomes. So I'll repeat, HGP is an ambitious international project designed to understand human genome, that is, to identify and locate 35 to 40,000 genes present on 23 chromosomes, or you can tell 23 pairs of chromosome maintained in the nucleus of a human cell. Or you have one more definition. What is that? It's an ambitious international project which deals with molecular organization of human genome, their information, and the gene product they encode. That is, we want to know what is the molecular structure of genes. We want to know what does this genes contain and how they are going to help in producing a particular product. So thus any of these two definitions can be used for HGP. Now let us come for the next part of the topic, that is Human Genome Project history, when it was started and when it was completed. HGP was started on October 1990 in USA and was coordinated by US Department of Energy and National Institute of Health, NIH, and also another country, that is UK. Wellcome Trust from UK became a major partner. In other words, Human Genome Project, initially it was started by coordination of USA and UK. But other countries also joined this. Who are they? That is Japan, France, Germany, China, and others. Remember, India did not take part in this. I'll tell you later on why. Now, this Human Genome Project, when it was initially started, it was a 13-year project and was estimated to be completed in 2003. So this is the background of Human Genome Project. Now, let us proceed for the next thing. Human Genome Project was referred as a mega project. Why do we call Human Genome Project as a mega project? One thing is because there are two important magnitudes and there are requirements. So let us see what are they. Human Genome Project was approximately known to have 3 into 10 to the power 9 base pairs, and the cost of what can say mapping this 3 into 10 to the power 9 base pair was US dollar 3 per base pair. That is, to locate one base pair, US American dollar $3 was used, and when we calculated the total amount required for pairing 3 into 10 to the power 9 base pair, it was approximately 9 billion American dollars. So this is the first important reason to tell why HGP is a mega project. That is, the amount, the cost invested on this is nearly very high. Second important reason is, so when we estimated the project cost and if the project would have been successful, and when we get the information, the book required is, that is, if each page book contained 1,000 letters and each book contained 1,000 pages, nearly 3,300 books were required to store information of DNA sequence from a single human cell. Imagine what is the magnitude of the books. So for one human cell to note down the gene sequences, 3,300 books were required. So you can imagine what is the highest okay, so what is the highest magnitude of this project. That's why we call this project as a major what is that, mega project. The third important reason is the enormous amount of the data expected to be generated also necessitated the use of high computational devices for data storage, retrieval, and analysis. That is, storing information in 3,300 books was not easy. Even if it is done, thing is, we want the information in a fraction of second. Just when we click a button, the information should be available. So therefore, this project gave an additional work to IT people. That is, they had to develop high speed computers for storing the data, for what can say, retrieving it, and for carrying out analysis. So therefore, this is the other important requirement that Human Genome Project gave rise to. Hence, IT people started working on this. So therefore, apart from what can say, molecular biologists, even IT people were involved in human genome project. So this is the first important great requirement, that is development of computers with high data storage and analysis was the prime requirement. Then the last thing is, HGP was closely associated with rapid development of a new area in biology called as bioinformatics. That is, yes, by starting with Human Genome Project, a new branch in biology, that is bioinformatics. What is this bioinformatics? Analyzing the human genome with the help of IT professionals is what we call it as bioinformatics. And at present, this is the very important branch of biology that is working on Human Genome Project. So therefore, because of this four important reasons, that is two magnitudes and two requirements. Two magnitude is the amount, more huge amount was invested. Second important magnitude was more number of books, that is 3,300 books were required. These are the two highest magnitude, and this project led for development of what we can call, the requirement was, there was a need for development of highly sophisticated computers. Then second thing was, it also led for the development of a new branch of biology called as bioinformatics. So because of this two magnitude and two requirement, Human Genome Project is said to be as a mega project. So now let us move on to the next important part, that is goals of Human Genome Project. So what was the goal set up when we started with this HGP? What was the need to carry out this? So let us try to, and this is very very important. Remember, for 5 marks, a very very important questions for you. List out the goals of Human Genome Project. So let me come for the goals of the HGP. The first goal is, yes, when we started, the most important was to identify approximately 20,000 to 25,000 genes in human DNA. That is, at that time when this project was started, it was believed that human DNA contains 20 to 25,000 genes. So therefore, to identify all this, Human Genome Project was started. Second thing was to determine the sequences of 3 billion chemical base pair that make up human DNA. That is, in this 25,000 genes, which was approximate, they estimated that in this 25,000 genes, there was 3 billion nitrogenous base pairs. So, how they were arranged? What is the pattern of arrangement of this base pair? So, determine that this was the goal. Then third thing is store this information in databases. Yes, I told you, if we had to store the information, we required 3,300 books of 1,000 pages, and in each page 1,000 words. But this was very difficult because you cannot keep on searching the pages if the information is required. As I told you, on just one tap you should get the information. So for that, what can say, we had to develop better computer. So we have to develop better computers, and that information should be stored. So the goal was to store this information of UHGP. Then the fourth thing is improve tools for data analysis. I told you storing 3,300 pages, storing 3,300 books information on computer was not very easy. So therefore, they had to develop what we call today as software. So software has to be developed and it should work according to our need. So therefore, to improve the tools for data analysis was the another important goal. Transfer the related technologies to other sectors such as industries. Yes, this HGP is required. Might be, for example, to identify genetic disorders, might be to treat genetic disorder, or to know the transfer of characters, and to develop what can say treatment methodologies. So for those industries, everything cannot be given. What part is required? So that information has to be transferred to the industries. So therefore, that was the idea. That is, if we get the information about human genes, so we can make use of this information or this knowledge for what can say making human life better. So you can produce better products. For example, you take even your food, nutrition. So we can develop better nutrition according to our requirements. So therefore, some industries needed this. So the main goal was after developing this HGP, we can transfer the information to industries. Then address the ethical, legal, and social issues that may arise from the project. What is that? Ethical, that is religious problem, legal, any public issues, or society issues. What is that? Thing is, I already told you in the beginning, India did not take part in the HGP. Why? It was an ethical issue. That is, Indian religion did not permit to develop HGP. So therefore, India did not take part in this. Legal. Okay, after if you come to develop HGP, if you come to know that your genes are recessive, they are bad, you have got genes concerned with diseases, then let us say many of the what we can call policy makers, or let us say for example LIC, they may not provide you the LIC bond. So at that time, how to over these issues may arise. Or let us say for example, initially you know that Europeans were following apartheid policy. So now if they come to know that their gene is supreme and the black people genes are inferior, don't you feel the difference? So then there will be a clash between the society. So to address these issues. Social issues. Yes, among the society there are many regions, there are many type of people. So if they have, if they come to know that their genes are superior or inferior, that may give rise to a problem in society. So therefore to address all this, in case if genes are what can say known to be inferior, superior, or there are positive and negative things about the genes in a human being. So what problems may arise and how we are going to handle this. So therefore that was the other important goal that was set up by Human Genome Project. So this is with regarding to goals. So in the next part of this slide, we will be talking about methodologies. So let us proceed with